Go ahead.
Okay, we're all right?
Mm-hmm.
Okay, well, welcome to the Good Manufacturing Practices and Quality Assurance session of biotechnology. I've been associated with the program for a while now. In fact, I actually worked together and suggested some of the criteria for the original curriculum. So I worked with Phyllis and Kuntabi, and yeah, we put together some original ideas, and I think really it's one of the best programs I've seen in the whole state of Ohio. So I think you've all made a really good decision to be part of the program. I think it's really works, and I'm here to say that, you know, in fact, I have one student now, Jenny Smith, who was one of Susan's students, and she started with us as an intern, and then she moved to part-time, and now she's full-time. So she's a full-time permanent employee, and it can happen for just about anyone. And it's amazing, you know, how far you can go with what you do. So that's really the message that I want to give to you today. I'm not going to spend a whole lot of time, you know, filling you in on my background. If you want to know who I am, this is it right here. Basically, I'm a microbiologist, Ohio State University. Then I went into the field of quality, quality assurance, specifically certified quality auditor, certified quality improvement associate. I'm also a certified nuclear medicine technologist. My advice to all of you as students, and all of us are students our whole life, otherwise it can be pretty boring. As a student, you need to be open to new opportunities, any opportunity, quite honestly. I would have never thought I would be a nuclear medicine technologist. That's not what I studied at The Ohio State University. I studied microbiology. But when I started interviewing, one of the first questions was, well, what job did you have that has anything to do with microbiology? I'd had jobs since I was 16, but none of them were directly related to microbiology. So it's like, well, plan B. So that's when I became a certified nuclear medicine technologist. I figured that way I could get into hospitals, get into healthcare, which is a hot area and will always be a hot area. So, I mean, a lot of times you have to zig or zag. You may not be able to take the path that you thought you could take. My husband's a mechanical engineer, and those people have a job before they leave campus, and it's in engineering. It's not in something they hadn't studied. So it just, you know, just open yourself up. You'd be amazed at how a lot of the things you do can end up contributing to your overall expertise. And there may just be a perfect position out there for you, especially at a smaller facility like the Rogeson where I work. You really need to have a lot of skills that go across a broad range of specialties. So when the organization is small, you're going to wear a lot of different hats. So that's what I would say. That's my advice. I'm sticking to it. It worked for me, so that's what I tell all of the students. In fact, right now we have a really great intern from Wright State, and he is in biomedical engineering. He's a junior, and I mean, it was just really strange. I met his mom at the post office. I go to the post office a lot because my whole family lives in another state, so I mail a lot of stuff. So I've been going there for years, over 10 years in Xenia. And she saw my Bob Evans ID. I had it on when I dropped something off at the post office, and she said, "Do you work at Bob Evans?" And I said, "Well, they're our partners in research." I said, "Yeah, we do work with them." She said, "Do they do internships there?" And I said, "Well, the Rogeson does internships. Bob Evans doesn't." I said, "But we do." And she said, "My son's in biomedical engineering, and could he drop his resume off?" Sure, no problem. So he came over and we met Tony, and he's just the greatest kid, and everyone is just basically fighting over him. Quality was the person who took the internship. Yeah, everyone else was like, "No, I don't really think we need an intern right now." Well, suddenly after everyone met Tony, they needed an intern. Because Tony is very bright. And Tony is enthusiastic, and he is willing to do anything. And that's the other lesson I have for all of you. Be bright, enthusiastic, and willing to do anything. Just open yourself up. Just because you've never done something before doesn't mean you shouldn't learn how to do that. I mean, it's really very interesting to learn what other people do as part of a job. Even things like incoming receiving. That might sound boring, but quite honestly, that's really where everything starts. You have to have quality incoming raw materials, or you're not going to have a quality finished product. So those are just a few little tidbits that I like to share with the students, and I appreciate Susan inviting us here each year to share those thoughts with you, because I think it's important to stay connected to what's actually happening in the real world, because it does change really quickly. Yes?
What do you do with products? Do you do like agricultural biotechnology?
No, actually we do diabetic research, and they allow us to harvest cells from the hog pancreas, which normally they would throw away at the sausage factory. It goes right in the dumpster. But we get to section out the pancreas and get the islet cells out and make our therapy, which is what I'm going to talk about today. So we have a diabetic therapy and a cancer therapy.
Hogs are like a lot similar.
They're extremely similar. Yes, the islet cell in the hog pancreas is one amino acid different from ours. So if your islets aren't working and you're diabetic, your body doesn't know how to process sugar, you can encapsulate hog islets, and they know exactly how to do it, even in a human body. So yeah, the key is that we encapsulate the cells in a gel bead, and that allows the body not to recognize those as a foreign cell, and so it doesn't reject the cell at all. The patients don't have to be on any kind of immunosuppressive therapy. So I'm going to pass these around. This is what we do at the Rogosin. This is the best place to start, because this is what gets us all excited at the Rogosin. In fact, this morning we had a patient re-implant, and that makes everybody's week. We are giving quality of life to stage 4 cancer patients that have failed radiation and chemotherapy. And we implant these macrobeads into their peritoneal cavity. And the macrobeads are living cells, and when you encapsulate them in a small gel bead, and if you hold these up to the light, you can see the colony-forming units inside the beads. So just pass those around to the last row there.
Are these the cells like inside of the bead?
Yes, those are growing cells. They're forming colonies. So we do cell culture, which I'm sure a lot of you have got a chance to experience in your classes. We do cell culture. We harvest the cells for cancer, and then we encapsulate them in the double-coated gel bead. The gel is a highly purified seaweed agarose, which the patient has no reaction to either. It's pretty much non-allergenic. So I'm going to go through a little bit more about the beads as I go through the presentation here, but I like to start by giving you something physical to look at. But as I say, today we had a re-implant of a patient. Generally, we will put in anywhere from 400 to 700 macrobeads in the patient's peritoneal cavity. There's a lot of open space in there. We make a very small incision, and we use an endoscope, and we funnel them down through with an endoscope. And usually they stay highly active for about 6 months, and then at the end of 6 months, the patient, depending on their condition and whether or not they would like to, can have a second implant. The majority of our patients, we're over 200 patients now, have all had at least 2 implants. Our star patient has had 5. She's an executive in France and has been able to, remain active as a CEO and work full time, thanks to the macrobeads, because she was diagnosed with ovarian cancer. Wow. So she is by far the star, but all of our patients are stars to us. And because they've chosen to participate in the human clinical trial, they're making a contribution not only to science, but a contribution to other patients that have terminal cancer, because this gives them another option. And what we're hoping is very soon the Food and Drug Administration will allow us to do implants in a stage 3 or a stage 2 or even a stage 1 cancer patient, because we don't know what the macrobide will do if we can get it in there earlier. It might even be more effective. But right now, FDA says stage 4 only. And you can't go, you don't go against anything the FDA says. So that's the program for right now. We're in phase 2B. We're going to be doing a series of colon cancer patients and focus on the colon cancer, but we've treated a variety of different cancers: pancreatic, ovarian, lung cancer, liver cancer, breast cancer. So we have done a variety, but we are focusing on the colon cancer in this next section of patients. We summarize all the data, then we meet with the FDA and hopefully convince them that, you know, our project is worthy of being granted phase 3 human clinical trials. And hopefully, as I say, we want to get in at an earlier stage so that we can help our patients. Okay, let's just get into the presentation here, so hopefully it'll start to make some sense for you. The Xenia division is located right out here in the industrial park at the intersection of 68 and 35 in Xenia. And we're right next door to the Bob Evans facility. The Bob Evans group is incredibly generous. They've given us the entire second floor of their abattoir, which is French for slaughterhouse. It sounds a lot better though. They have given us the entire second floor so that we can harvest our islet cells from the hog pancreas. And that we do several times a week, and we then encapsulate those, and we have those in incubators for usually anywhere from 8 to 16 weeks. It gives them a chance to mature and really become active. And then we can do the implants. Right now, the diabetes project is only approved for animal model work, so we cannot do any implants in humans yet. But we are working with the FDA to get that moved on through to human clinical trial. The statement that you see here, that is our quality policy at the Xenia division. Remarkable research today, advanced treatments tomorrow, quality improvement at every step along the way. So quality plays a huge part in everything we do, and when you're doing research and development, you have to be creative, because a lot of times you're going to run into things that there is no benchmark for it. I can't look it up in a book because nobody else makes a macrobide. It's not like a pharmaceutical capsule, a medication. It's totally different than that. It's a bioartificial device, really, and it's pretty unique. The passion of the Robeson Institute Xenia division is to bring novel therapeutic medical devices to the patient as quickly as possible. And historically, they've found